Regulation of protein activity. The regulation of protein activity refers to the control mechanisms that modulate the function, localization, and activity of proteins within cells. Protein activity regulation is essential for maintaining cellular homeostasis, responding to stimuli, and orchestrating various cellular processes. Here are some key aspects of protein activity regulation. First is post-translational modifications, PTMs. PTMs are covalent modifications that occur after protein synthesis and can profoundly influence protein activity. Some common PTMs include phosphorylation, acetylation, methylation, ubiquitination, sumoylation, and glycosylation. These modifications can affect protein stability, subcellular localization, enzymatic activity, protein-protein interactions, and protein recognition. Let's discuss protein-protein interactions. Proteins often interact with other proteins to form complexes, and these interactions can regulate their activity. Protein-protein interactions can involve the binding of an activator or inhibitor protein to the target protein, leading to conformational changes that either enhance or inhibit its function. Examples include enzyme-substrate interactions, protein cascades, and protein complexes involved in signal transduction. Another is allosteric regulation. Allosteric regulation occurs when the binding of a molecule to one site on a protein affects the activity of a distant site. Allosteric modulators can be activators or inhibitors and can induce conformational changes in the protein, leading to the modulation of its activity. Allosteric regulation is commonly observed in enzymes, ion channels, and receptors. Another is protein degradation. Proteins can be regulated through selective degradation by the ubiquitin proteasome system or autophagy. Ubiquitin ligases can target specific proteins for ubiquitination, marking them for degradation by the proteasome. Protein degradation can control the levels of specific proteins, terminate protein activity, and regulate cellular processes such as cell cycle progression and immune response. Let's discuss subcellular localization. The localization of a protein within a cell can determine its activity. Proteins may be targeted to specific organelles, compartments, or cellular structures, allowing them to interact with specific substrates or modulators. The regulation of protein localization can occur through signal sequences, protein motifs, or interactions with other proteins or membranes. Now, let's discuss feedback mechanisms. Feedback mechanisms can regulate protein activity by sensing and responding to changes in the cellular environment. Feedback loops can involve the product of a biochemical pathway acting as an inhibitor or activator of an upstream enzyme, ensuring the maintenance of proper cellular homeostasis. And another is transcriptional regulation. While not directly regulating protein activity, transcriptional regulation plays a significant role in determining protein levels, which can subsequently impact protein activity. Another pathway is cellular signaling pathways. Protein activity can be regulated by signaling pathways triggered by various extracellular or intracellular signals. These pathways often involve the activation of specific protein kinases or phosphatases that phosphorylate or dephosphorylate target proteins, respectively, leading to changes in their activity. Examples include the protein kinase PKO pathway and the mitogen-activated protein kinase MAP pathway. The regulation of protein activity is a complex and highly coordinated process that involves multiple levels of control. The interplay between post-translational modifications, protein-protein interactions, signaling pathways, allosteric regulation, cellular localization, and feedback mechanisms ensures the precise regulation of protein function in response to cellular needs and environmental cues. Thank you for watching, and don't forget to subscribe, like and share.